Yeah, my name is Nils Peder Willassen, and I'm from the Arctic University of Norway, and that is far north. It's located in Tromsø. So we are far south now compared to Tromsø, but it's still cold. I want to tell you a little bit about metagenomics, and but first I want to say some words about Elixir, then a little bit about the background, what can we do with metagenomics, and most important, the different steps in metagenomics we have to think about before we start, and the main challenges that we are facing today. So Aja was talking about Elixir, and this is a huge network trying to build up bioinformatics throughout Europe. So this is a collaboration between 20 members and one actually observer. But the idea is to link all the national nodes together so we can build the infrastructure to cope with all the data that we are producing each day. So this is the member countries, and there is more than 140 institutions involved in this network and more than 400 scientists. So it's a huge network. So what we are trying to do in Elixir is to build up platforms, the data platform, how to cope with the data, how to store it, the databases. We need standards so we can use the data. We need tools. And of course, we need access to compute to actually crunch all the data to get out the results, and of course, training. In addition to these technical platforms, we have also involved 4 different user communities: the marine metagenomics, crop and forest plants, human data, and rare diseases. So this is the whole operation. And as I said, there are many institutions involved and also many countries involved in this. So I will talk about a little bit of marine metagenomics before we continue on genomics in general. So the marine one, so our mission is actually to develop a sustainable infrastructure to enhance research and innovation. That is the main goal of the marine metagenomics use case. So that is led by Rob Finn at the EBI and myself. So the action is actually to try to develop standards for the marine domain. It's very important to have good standards. We will talk about this in a lecture tomorrow. We need to develop databases that is specific to the domain that you are working in. In this case, we are working on databases for the marine metagenomics. There will be a lecture on that also tomorrow. We need to implement tools and pipelines, also to make gold standards, best practice for metagenomic analysis, how to do it, how to analyze the data. And of course, we need also to have good search engines so we can actually get the data that has been published. If you want to compare it, you need the data from databases and how to find it easily. And we need also to develop and deliver training for the scientists, for the students, and so on, to learn how to use the data resources that is available, how to use the tools and pipelines. So, what is metagenomics? There are thousands of definitions of that, but I like to use the simple one. It's actually the study of genetic material recovered directly from environmental samples. And the idea behind that is, of course, that you want to explore the relation between the microbes and the habitats. So that could be marine, it could be human intestine, it could be whatsoever. But you need to know what actually do you want to look at. So this is a slide I made many years ago. I don't remember it now, but at least 15 years ago I started on this. So the typical way to look at habitat or environment is, of course, what we did before was cultivation. So we took samples, we plated it out, we picked colonies, we made culture of that, we isolated the DNA, and of course we sequenced the genome. So in principle, from a diverse environment, you have access to approximately 1%. For the human intestine now, I think it's 35%, but in a complex environment, it's very low. Then we have the metagenomic side of it. You just sequence everything. So in principle, you have access to 100%. But that is in principle. I will show you later why that is probably not correct. In addition to that, you can isolate RNA, protein, metabolites, and also analyze those, transcriptomics, proteomics, metabolomics, and so on. And of course, we have the genomics and the metagenomics, we have the multiomics, and we have the metaomics, and we have everything in between. So it's a lot of omics inside this. So when I'm talking about multiomics, I'm talking about using single cells, and if it is metaomics, then you use genomic samples. So what can we do? Of course, if you are using markers like 16S, you can look at identify new species, you can identify what's there. You can look at the diversity in the community, and you can compare it with different communities. You can look at time series, you can look at different states. It's a lot of possibilities to do comparison between two samples or many samples at the same time. But we have a problem here, and that is classification of eukaryotes, unicellular microorganisms, especially fungi, protists, and also viruses. We are far beyond where we should be. Prokaryotes, starting to get it up and running well, but this microorganism is far beyond where we should be. We can do functional assignment of the protein coding sequences, of course. We can try to reconstruct pathway in the communities. We can compare in the different states or different sites or whatsoever. And we can look for novel sequences, doing bioprospecting, looking for novel enzymes, for example. And we can look at new pathways. So there is a lot of possibilities to analyze based on the protein sequences. So what are the applications? If you look into the literature, you can find a lot of applications. Plant microbe interactions, for example. Try to improve crop yield. Monitoring and optimizing wastewater treatment. Bioprospecting, as I mentioned. looking for novel enzymes and compounds, functional and ecology of the oceans, like the Tara Ocean Expedition, Malaspina expedition. You have also the Open Sampling Day. Huge projects, worldwide projects. Development of functional foods for farmed animals. Monitoring pathogens, monitoring pollutions, and of course, surveillance and treatment of disease. So there is a lot of interest in metagenomics now. So this is just a few examples of what they are using the metagenomics for. So the big picture is very simple. You have heard how to sample today, how to generate data, and we now are facing this to start to process data and to analyze it. But before we start on processing and analyzing, I will just start from the beginning again with the sampling, and even before that. So the first thing we have to do is the plan. We need a plan how to do it. So the project plan, we need to describe the aim or the goals of the project. We have to keep that in mind all the time. We need to set up the experiments, experimental design. We need also to develop a data management plan, because we will generate a lot of data, and how to handle this data. We cannot have it in Excel sheets, and we cannot store it in the drawer. We need to store it somewhere, and we need a plan for that. So the plan is very important. That is where we start. The next one is, of course, the sampling. As we already heard, what, where, when, and how to sample is the most important. Do we have the required equipment for doing the sampling? If you are sampling in the open ocean down to 4,000 meters, do we have the equipment to do that? Or at the bottom floor? We need, in some cases, enrichment, filtering. We were talking about in the coffee break, virus or vironome. Then we need to filter out that we just are looking at the viruses. Do we have the equipment to do that? And how to do it? How to store the samples when we have sampled it means a lot. And of course, what we also need to know at a very early stage is the diversity of the sample. Do we have any idea of the diversity of the sample? The next step, as we already heard, is isolation of DNA. And it's not straightforward either. So how to lyse the cells and isolate the DNA? We need probably to remove major contaminants in the very early stage. At least with the polluted samples, oil polluted samples, for example, it's totally mess if you try to isolate the DNA from that. How to lyse the microorganisms? We know that most of the technologies and the methods that we have is optimized for gram-negative. Gram-positive is harder. Fungi is not so easy. But if we want to capture everything, we need to optimize these technologies and methods. So we are actually capturing everything and not just a part of the microbiome. Viruses, We probably need to isolate those before we actually add it to the rest of the samples. So it's a lot of thinking about how to isolate the DNA from the samples. And of course, what is really important before we go further is the quantity and quality of DNA. We need a high quality, and of course, we need enough DNA to sequence to make these libraries before we start sequencing. Sequencing, we have heard a lot of the different technologies to use. So Illumina, Ion Torrent, PacBio, Oxford Nanopores. Some is used a lot, some are just in the beginning, but still there is a lot of different technologies to take care of. I have not mentioned 454 because they are closing down everything. So, but still there is a lot of data sets in the databases that is based on the 454. So what kind of sequencing mode do we want to use? Single reads, paired ends? What about the read length? We heard that the longer the better. And of course, but it's a choice that we have to take. And in many cases, we have just to use the machine that we have at hand. We cannot say that we want to use PacBio, for example, if there is no PacBio nearby. If it is a MySeq, you more probably have to use it. So, and replicates. You have heard earlier about how many replicates you need, biological, technical. So you need to take this into account also very early. Sequencing depth. How much should we sequence of each of the samples? And that is why it is important to know the diversity in the beginning. If you don't know the diversity, how deep should you actually sequence a sample? And of course, price. I will give you some examples, or just one example, to show you that it's not cheap to do metagenomics if you want to have a good sequencing depth, at least. So this is from the marine domain, just show you some numbers. So the number of species we don't know, but it could be billions in a sediment. And for the bacteria, and the genome size is approximately 5 megabases. Viruses, we don't either, but probably millions of different species. The protist is a little bit lower, but still we have no clue. More than 300,000. This numbers is taken from the literature, so hopefully they are correct. Fungi, more than 150,000 different species. But you can see here, viruses, small genome, bacteria up to 12 megabases, average approximately 5. Fungi, a little bit bigger. The protist can be huge. If you compare it to the human, that is approximately here, so you can see the protist is gigantic. And that is not easy to handle all of this variety of sizes of the genomes when we are doing metagenomes. So just to give you an example, if you just calculate, if you have a sediment, seawater or sediment, so if you take the average, you end up with approximately 5 terabases per gram of DNA. And that is quite a huge amount of DNA. Of course, it's a little bit the number of viruses is quite amazing, so you end up with approximately 500 terabases per gram, and so on. So you can, a simple calculation if you take the average numbers. And of course, metagenomics is not about only bacteria, but it's about all of these groups of cells. But just to give you one example of, if you take the sediment and want to sequence everything, how long will it take and what is the cost of it? So if you're using the MySeq system and you will try to cover it just one time, we know that the output from the MySeq is about 15 gigabases per run. And if you are using 2 times 300 base pair, and it will take approximately 67 hours from you starting the library to you have your results out, and you can just calculate it, it will take 330 runs to sequence everything one time. 2.5 years. So you can see that this is not possible to do. And the price, yeah. So if you would take, as we do with a single genome, approximately 100 time coverage, yeah, 250 years. And 66 million euros. So yeah, we shouldn't do that. But it just says something about we need to plan it carefully, what we are doing from the beginning. You need to start to calculate. You need to know the diversity in your sample and the sequencing depth that you want to have. Otherwise, you end up with these numbers and it's not possible. And then you have to go back again and start from the beginning and rethink about what is the objective of and what is the goal that I want to achieve in doing such a project. So we need to think very carefully about what we are doing. But of course, we could choose a high throughput machine to do that. We will get down the number of years to do the experiment. But of course, if you are reducing the length of it, then you have other problems afterwards when you are starting to analyze it. So the next is, of course, processing and annotation. First of all, you need actually to transfer your data from the sequencing machine to somewhere where you can store it safely. And that is also a problem today, because its data sets is huge. And it's a problem to actually transfer it from the machine to a server. In many cases, you need actually more time to transfer the data than to produce it. So that is also a big problem. The next is, of course, which tools and pipelines to use to do the processing and annotation of the sequencing reads. What kind of reference databases do you want to use? Availability and scalability. You can use nice tools on a small data set, but they don't scale, so you cannot use it on huge data sets. And of course, this requires that you have access to high-performance computing. And you need to store the processed data in some way also. Not only the raw data, but also the processed data. So after you have processed the data, you have mapped the 16S and the protein sequences. you have the functional assignment, all of those, then you can start to think about the next step. But this is just to show you our typical pipeline for metagenomics, where you have different tools, you have different programs, and you have different reference databases that you use all the time. So that is why this importance of reference databases, what to use and how to use those. Some of these programs are very easy. It scales very well if you have huge samples. Other ones doesn't. So there are always some bottlenecks that you have to think very careful about. Espen will tell you more about MetaPipe and how it's working, and you will, during this hands-on, use this pipeline for analysis. So the final step is, of course, do the analysis. Who is there, what can they do, and how do you do it? But what should you do? Taxonomic classification or functional analysis, or both? Do you have the tools to do that? And also the reference databases that you need to do the analysis? You need to publish your results. And of course, that requires that you have to deposit the sequences to ENA, for example, or other public databases, archives. And you need to follow a minimum sets of information from the planning, actually, and throughout the whole process. We will come back to how we are working with standards and how to standardize the whole process. So what is important is, of course, to have the project plan. You need to do the design of experiments. You need to have a data management plan. You need also to make a standard operational procedure, so you are doing exactly the same things every time you do a sampling, you do the isolation and everything. Otherwise, it's impossible to compare the samples. And you have to follow it also. You need also to track the data, what is called provenance data or metadata or contextual data. So you need to describe every step. I will tell you more about this metadata tomorrow when I will talk about how we are making a new reference databases and how important that is. We need also to develop gold standards or best practice. Probably not gold, but silver standards. It's hard to make gold standards, but at least try to make it as best as possible. And that means that you have to benchmark tools and pipelines. And you have to know exactly what is going on in each of the different steps in a pipeline. What are the problems with merging sequencing reads? What are the problems with filtering? What effect does it have on the end results? So you have to go through all of these steps and try to figure out what it means. So what are the main challenges that we see today and that we are facing? One is, of course, the number of sizes of projects are increasing. Before it was just a few, now they have time series over years. Every month, for example, take a sample and just follow that over many, many years. The size, of course, is increasing because new technology is getting cheaper and cheaper to do it. The problem is it's getting cheaper to do the sequencing, but the analysis gets more and more expensive. You need scalable tools and pipelines, and you need high-performance computing. Otherwise, you have no chance to do large projects. Short sequencing reads is very hard to annotate. If you have 300 base pair, it's much easier to annotate than you have 150, for example. So that is a problem. But we don't know the future, but we just hope it gets longer and longer reads. And as always, it's very few hits in the reference databases. So we cannot annotate at all. So this is just an example from a sediment sample in the Arctic Ocean. So you can easily see that around 80–90% of the rRNA from archaea we cannot annotate at all. It's a little bit better with the bacteria. It's around 45–50%. We know that this is RNA from bacteria, but we cannot annotate it because there are nothing like it in the database. And of course, there's a lot of unclassified also. That means that we need domain-specific databases to handle this and to get more and more actually genome sequence that we can put into the databases. And we need to do more annotation of metagenome samples so we can use that also in the databases. And of course, we should be aware of that the results depends heavily on the tools that we are using, the algorithms, and of course the reference databases. So not only do exactly the same when you are sampling, isolating the DNA, but also when you're doing analysis. Use the same tools, use the same version of the database. Because if you are using a totally different database version of that, it will give totally different results. And also, if there is an upgrade of the version of the tool, it will also give different results. So you need to use exactly the same. And that is hard to find, this information. If you will go out in a database and you will compare your sample to another one that is deposited in an archive, you cannot find any information about that. So if you want to do it properly, you have to rerun the samples that you find in the different archives. So, and of course, as I said, the description of data, also the metadata, is often lacking, and that is something that we need to do something about. But I will come back to that also tomorrow in a lecture. So by that, I think I'll just stop for now. Any questions? Do we have a microphone? The small microphone.
Thank you. So it's about the use of old genomes versus the use of genes which are representative of those genomes. So could you comment on that, especially in the case of human biome?
Yes, the idea is of course to we need to sequence as many genomes as possible and put these into databases. Not only the genome, but of course the coding sequences. And you can use that again to annotate your metagenome samples. So the better the databases, the better the results you will get. So to sequence genomes still is very, very important. The more the better, and the more exact result you will get. Not only from taxonomic classification, but also to inferring, for example, different pathways and so on. It's the more the better, of course. But in many cases you don't have the whole genome. So then you have to use marker, for example. And of course it's also, if you want to use BLAST against it, is of course the bigger the database is, the longer it takes. So in many cases, yeah, it's okay to use specific markers, yeah. But we need actually all the microorganisms in the human microbiome to be sure that everything we annotate in a metagenome is correct, or as correct as we can.